At the National Xenopus Resource, we have over 10,000 frogs. We breed, maintain, and distribute frogs to the researchers.
The African clawed frog, Xenopus laevis, is studied by researchers around the world who use it to answer all kinds of questions in biology.
What makes Xenopus unique is that they can produce up to 5,000 eggs. Huge eggs. The eggs are 1 millimeter in diameter, and they're extremely hardy embryos.
This frog's path to biology superstar has a surprising beginning.
The story of Xenopus starts with the pregnancy test.
I had not, before you mentioned it, heard the story about the frog and the pregnancy test. My name is Anshanta Jenkins, and I am a board-certified OBGYN. Pregnancy is the process by which an egg has been fertilized and is now growing into a being.
Eggs are produced, stored, and released from organs called ovaries.
Ovulation is when an ovary releases an egg.
After ovulation, the egg is transported through a tube called the oviduct, where it can be fertilized by a sperm. If it's fertilized, the egg starts to divide and may implant into the lining of the uterus.
Implantation is the process whereby the fertilized egg will burrow or go into the lining.
As the fertilized egg divides, the cells that will become the placenta, the organ that supplies oxygen and nutrients to the fetus, release hormones, molecules that provide signals to other cells. And one hormone is especially important.
Human chorionic gonadotropin, HCG, is the hormone that the body produces when pregnancy is in existence in the body. It's going to support the pregnancy until the placenta is up and running and things are in place to continue the pregnancy.
HCG levels increase for the first 10 weeks of pregnancy and then start to decline. The hormone can be detected in the blood and urine starting around 10 days after fertilization. Thousands of years ago, long before HCG had been discovered, people from many cultures knew there was something special about a pregnant person's urine. For example, when they put urine on certain plants, the plant changed only if the person was pregnant. Then, scientists discovered a new test. The year was 1929. British zoologist Lancelot Hogben was doing research in South Africa.
He got interested in the local species there, Xenopus laevis.
Back in Britain, Hogben and his students injected Xenopus with human urine. The urine of a pregnant person caused a surprising result.
What they found with Xenopus was that you could inject the urine into the back of the frog and she would lay eggs.
Within 15 hours, if the person was pregnant, the injected frog would produce eggs. If the person was not pregnant, no eggs. It was a faster pregnancy test than anything discovered before.
And very quickly, by the late 30s, they were using Xenopus as the main pregnancy test in Britain. And then they started exporting these frogs all over the world. They came to America, so everybody started using it.
By the 1960s, knowledge that HCG in a pregnant person's urine caused Xenopus to lay eggs opened the door to simpler tests.
The way that modern pregnancy tests work is that they are going to take the urine, just a small amount of it, to check for pregnancy. And basically, they're basing that on the HCG hormone.
Modern pregnancy tests consist of a test strip with 3 sets of antibodies, proteins that bind to specific substances. The first set of antibodies are mobile and contain a dye. The second and third set of antibodies don't have a dye and are stuck to the test strip. When urine is added to the test strip, the first set of antibodies will bind to any HCG molecules present. If there's no HCG, these antibodies won't bind to anything. As urine moves up the test strip, it carries these mobile antibodies to the second set of antibodies on the strip, which also bind to HCG. The dye creates a visible line. If there is no HCG to bind to, there's no line. The urine continues moving up the test strip, carrying any mobile antibodies without HCG. These mobile antibodies bind to the third set of antibodies on the strip, and the dye creates another visible line, a control line, that tells us the tests worked properly.
2 lines is yes, 1 line is no.
Their popularity as pregnancy tests brought Xenopus to the attention of researchers worldwide, who quickly started using the frogs as model organisms for studying how eggs form and mature, and how embryos develop.
And so you can easily study the same process in a frog and then make applications to humans. It's not going to be perfect, but it gives you a platform or a foundation.
Everything is building on the other, right? Whether it's mathematics or whether it's science. Information that they knew many years ago, we just keep adding on to the information, and that will help us better diagnose, better care for patients, come up with new ideas. And we've come a long way, baby, because now you can go in the store and buy something that started out with a frog.